Hello, welcome. My name is Grace, and this is my partner, Beer. We did our enzyme project on cocaine esterase, and here it is, the main feature of this slide.
Ta-da! The organization of this presentation will be as follows. We will first start with the general information to know about our enzyme cocaine esterase. After we present to you the general information on our enzyme, we will then introduce to you our paper's aim and how they conducted the experiment by pointing out key materials and methods. Following presenting our materials and methods, we will discuss the results of the paper. Lastly, we will wrap up our presentation with the takeaway slide, concluding with the key points you need to know about our enzyme and our paper. So general information. What is cocaine esterase? Cocaine esterase is an enzyme that comes from the bacterial species Rhodococcus. The type of enzyme is found in the rhizosphere soil surrounding the cocaine plant, also known as erythroxylum coca plant. The way that this enzyme and all other enzymes are classified is by using something referred to as an enzyme commission number, or the EC number. This numerical classification is a way to distinguish a particular enzyme based on a chemical reaction that enzyme catalyzes. So for enzyme esterase, its EC number is 31184. The 3 indicates the kind of enzyme it is, a hydrolase. A hydrolase enzyme is an enzyme that cleaves a covalent bond using water. The covalent bond cocaine esterase cleaves is an ester bond, indicated by the second number 1. The next number 1 indicates that with the cleaving of ester bonds, cocaine esterase is able to hydrolyze it into alcohols and carboxylic esters by being a specific type of hydrolase enzyme, a carboxylic ester hydrolase enzyme. The 84 states the actual name of the enzyme. So the paper we decided to look at was a biochemical characterization and structural analysis of a highly proficient cocaine esterase written by Turner et al. In 2002. This paper's main objective was to understand the structure of cocaine esterase's active site and understand the enzyme kinetics to further improve upon grasping the implication of its mechanisms using both wild-type cocaine E and other mutants of this enzyme. So here are terms to know. Here are terms we will be discussing in our paper. You will hear the terms interchangeably.
Here I'm going to discuss the methodology that the paper described in the research. The following procedures were conducted: cloning and protein expression, crystallization, mutagenesis, kinetic measurements, and finally urea denaturization. First was the cloning protein expression. This involved the production of the cocaine binding antibody, and then cocaine was subcloned to cocaine enzyme was subcloned to study the sequence of interest, and the soluble enzyme cocaine esterase was produced. Cocaine esterase was 99% pure through the use of iGross chromatography. And then we have crystallization, which gave rise to the crystal structure of the enzyme. And this involved the Y44F and S1178 mutants. These mutant sources were refined in CNS using the native enzyme structure as a starting model. The enzyme's purity was determined once again to be around 99% by dielectrophoresis stained with massive blue. Then we have mutagenesis. The quick change site directed mutagenesis kit was used with the standard desalted primers to mutate several codons in the enzyme's sequence. And then the desired mutation was confirmed by DNA sequencing. Then the kinetic measurements. So wild type and mutant enzyme concentrations were determined using the massive plus protein assay reagent kit with BSA as a standard. The initial rates B0 were determined by the following change in absorbance at 240 nanometers in the SpectraMax plus 384 UV plate reader. Initial rates were fit to the Michaelis-Menten equation, which from there the cat and Km were determined. The kinetic measurement overall validated the proposed mechanism and revealed the relative contributions of active site residues toward substrate recognition and catalysis. Finally, urea denaturization. So first, fluorescence was measured at room temperature using the SpectraMax Gemini spectrophlorometer plate reader. The final enzyme concentration was determined to be 1.5 micromolars. And then circular dichronism special was also collected on a AVIV 60DS polar meter, and the final enzyme concentration was determined to be 7 micromolar. Both of these procedures data was fit to a double sigmoidal plot, and the studies of cocaine esterase by fluorescence and circular dichronism show two unfolding transitions.
And so now for the results. The cocaine esterase enzyme is a globular protein consisting of 574 amino acids. Its respective molecular weight is around 65,000 Daltons. The globular protein is a mixture of beta sheets and alpha helices, subdivided into 3 domains: domain 1, domain 2, and domain 3. Domain 1 has a conical hydrolase fold composed of repeated beta alpha beta motifs. The repeated alpha beta alpha motifs create a central 10 standard beta sheet surrounded by alpha helices. The structure of domain 1 is predominantly running parallel and capped on the C-terminal edge by domain 2 on the beta folds of 6 and 7. Domain 2 is a helical structure composed of 7 helices that is packed anti-parallel. The last domain, domain 3, is composed of beta sheets with a beta barrel-like core formed by anti-parallel beta sheets. The crystal structure of cocaine E was solved by multi-anomalose dispersion known as MAD method.
Now, understanding mutations in active site. There was 12 mutants that were generated to validate the mechanism of cocaine esterase. These mutants do not follow Michaelis metakinetics because the mutants used required the amount of enzyme to exceed the substrate concentration. The 2 most important residues that play a major role in the binding pocket of cocaine esterase are S117 and Y44. The function of these residues are shown by their mutated crystallized structure. So, a serine residue. The active site is located at the interface. So the active site of the enzyme is located at the interface of all 3 domains. Within the active site is a prototypical catalytic triad, which this catalytic residue is a part of, along with 2 other ones. The serine amino acid residue is located in the nucleophilic elbow, which is a hallmark of the cocaine esterase folding pattern. The mutant validates the mechanism proposed by the crystal structure and shows that the serine residue is responsible for both the substrate recognition and catalysis, which is evident through the 72-fold increase in Km. And then we have the tyrosinase mutant. So the oxyanion hole is formed by the backbone of 2 tyrosine residues, which are efficient at the stabilization of the negatively charged tetrahedral intermediate in the reaction. Y4 plays a particular role in activating the substrate carbonyl. And the Km, however, was not changed because the mutant, but the tyrosinase mutant structure may serve as a model for the native enzyme.
So to expand more on what occurs in the active site, we have 3 figures in the left, A being cocaine E, the wild type, B being this serine mutant, and C being the tyrosine mutant. So we see that cocaine E wild type has benzoate bound in the active site and a single water molecule indicated by the red sphere hydrogen bounded to the catalytic amino acid residues. While the serine mutant of cocaine E also binds to benzoate, it lacks a water molecule, causing the histidine residue to swing down and form a hydrogen bond with aspartic acid. The tyrosine mutant is the inverse of the serine mutant, where it does not have a benzoate in the active site, but does have 2 water molecules also indicated by the red sphere. The 2 water molecules are relatively weak and are not within reach of hydrogen bonding. Thus, it's potential hydrogen bond donor or acceptor from the protein. The reason why this mutant lacks benzoate in the active site is because of the deletion of a hydrogen bond. If the deletion was not present, the hydrogen bond was to bind the benzoate with the tyrosine residue. Furthermore, because of the lack of the hydrogen bond, the binding pocket and the side chain becomes more disordered, with the tyrosine rotating a degree away from the binding pocket and tryptophan being the only amino acid residue to be able to stabilize the oxyanion hole in the binding pocket. So the cocaine E mechanism proposed and conditions was by using the results from our paper. So we found that cocaine E hydrolyzes cocaine faster than any other cocaine esterase found because of its large K-cat at 7.8 seconds to the negative first and a low Km with at 640 nanometers. Also, we found out that its optimal pH is at 9 with both the serine and the tyrosine mutant giving rise to the cocaine E mechanism. Although it's a hydrolase enzyme, we found that its mechanism is very close to tryptophans or chymotrypsin, which is a protease enzyme.
So we found the overall reaction scheme of the enzyme using BRENDA database. So we did a Google search of the enzyme database and clicked on the first link. And then we used the search engine and typed in the common name and clicked on the appropriate EC number. And then you scroll down on the main page until you reach the reaction mechanism, and I click on the reaction diagram symbol to get a comprehensive look at the mechanism with the external links that provide additional information on the reactants involved in the reaction scheme. So this is the overall reaction scheme of our enzyme, and here is the substrate cocaine. So cocaine esterase cleaves cocaine at the benzyl ester bond on cocaine using water to release econine methyl ester and benzoic acid. So here is the mechanism in closer detail. So we have first step, which is alkylation. So the tyrosine residues on the far left form the oxyanion hole, and then aspartic 259 hydrogen binds to the histine 287, resulting in an increase of the histidine amino acid pKa. Histidine can then activate serine 117 as a nucleophile by deproteinization. Additionally, tyrosine residue can activate the substrate ester bond as an electrophile along with the main chain of tyrosinase 118 serine, and then the serine residue attacks the carbon of the substrate benzyl ester. A tetrahedral state is formed. The oxyanion hole stabilizes the charge on this transition state, and the tetrahedral intermediate collapses, acylating the serine residue with the benzyl group. And the second step, hydrolysis results in a dissociation of the cocaine benzyl ester, which is the first product, and the enzyme, in which water molecule acts as a nucleophile and makes a second tetrahedral intermediate. And finally, dealkylation, the second tetrahedral intermediate collapses and the free form of the enzyme is restored with a second product, which is benzoate. Finally, with the takeaway slide. So here is the takeaway of our here are the takeaways of our presentation. The cocaine esterase is a hydrolase enzyme with 3 domains, 1, 2, 3, and then cocaine esterase catalyzes the breakdown of cocaine into metabolite, ecogine, methyl, enzyme, and benzoic acid. Cocaine esterase is highly efficient at destroying the drug cocaine's potency.
So also a main takeaway is knowing the crystal structure of cocaine E that was revealed by our presentation. So we know now a serine carboxyl esterase exists, as well as a hydrophobic active site and an oxyanion hole formed by the amide backbone from the tyrosine residue. Also, because of the paper, now we know a better understanding of the proposed mechanism of the esterase using the active site mutations.